Hello everyone, welcome to SR classes. Today we are going to see a very interesting topic in pharmaceutical analysis called bioluminescence assay. Before starting, just imagine this. Scientists are able to measure the contamination and biological substances using light produced by the living organisms. Yes, light is used as a measuring tool in this technique. Let us understand by step by step. Before that, we are moving to the definitions. So, usually bioluminescence assay is a method in which measure the concentration of the substance based on the amount of light produced in a chemical reactions. So, simply more substance, more reactions. More reactions is more light is produced. So, more light is produced. It is measured by the detectors. This concentration which is the light emitted, which gives us the amount of analyte present in the samples. So moving on to the principle, this method works on the simple enzymatic reactions. Here a molecule of luciferin reacts with the oxygen. This luciferin is usually naturally occurs from the natural sources such as fireflies, jellyfishes. These are bioluminescence effects by the luciferin compound only. So here a molecule called luciferin reacts with the oxygen in the presence of enzyme called luciferase, and ATP provides the energy. So this reactions produces the light energy. So basically chemically energy is converted into the light energy and this light is measured. So reaction explanation, first we have luciferin. This luciferin, when we add it to the sample contains ATP. So initially it forms the luciferin ATP complex. This luciferin ATP complex, in the presence of oxidation, in the presence of oxygen, it undergo oxidation reactions. So it converted into the oxyluciferins, and oxygen is converted into carbon dioxide and ATP is converted into the AMP. So when luciferin is converted into the oxyluciferin, it emits radiations. So this emitted radiation is in form of photons, not as heat. So important point is the intensity of light is directly proportional to the amount of analyte present in the sample. So biochemical components. So here the basic components involved in this reactions. Now let's quickly understand the components involved. We have luciferin, which is the light emitting substances, and we have luciferase, this highly specific catalytic enzyme, and then ATPA act as the energy source. Oxygen is required for the oxidations, and magnesium ion usually stabilize the luciferin, and we have a final device called luminator used to measure the light. Usually this luminator contains the photomultiplier tube as a detector. So types of bioluminescence assay. So in this type of bioluminescence assay there are four types. First one is the ATP bioluminescence assay. So ATP usually present in the cells. So the cell which are viable, for example, if we have the aseptic area, if we want to check whether the room is sterile or not, or aseptic, we have swab cultures. In that swab we have number of viable cells. So in that viable cells, if we add luciferin, it undergo biochemical reactions. If it produce luciferin, oxyluciferin, and emit photons, it is directly detected by the detectors. So if the sterile room contains the viable cells, we can usually detect it by the ATP bioluminescence assay. And then second is the luciferase reporter gene assay. So usually luciferase is inserted into the plasmid genes, which is DNA replications we have used. This plasmid, when replicate the luciferates, so it is used to in gene expressions. Whether the plasmid is expressing the particular genes, we can detect it by using inserting the luciferase. And the third one is wholesale assay. So this wholesale assay is used in toxicity and drug testings. And then is fourth one is immunobioluminescence assay, used for the antigen and antibody detections. So moving on to the next topic, luminator. Now let us understand the instrument used called luminator. It is a device that detect light emitted from the reactions. So it is biochemical reactions which emits the photon, not in form of heat. These photons are detected by a photomultiplier tube, and the whole entire shape is called the setup is known as the luminator. So it has a photomultiplier tube which captures the light, and amplifies the signal, and display is as RLU, relative light units. So that moving on to the procedure. Now the procedure is very simple. So first we collect the sample. This sample is maybe a biological sample, an environmental sample, or any other sample. In that sample, we are adding the luciferin. So luciferin is we are adding to the sample, and then we are adding to the luciferase reagents. So this reaction start in the presence of ATP only. So light is produced, the illuminator detects the light, and we finally result in the relative light units. So how much of light is emitted is expressed in term of relative light units. So the concentration is directly proportional to the relative light units. So here we have the pharmaceutical applications. So we have several pharmaceutical applications. For example, in quality control. In quality control, we are detecting the microbial, whether it is present or not in the sterile injection or ophthalmic products. And clean room monitoring. As I said before, in clean room monitoring, we are taking the cells either to swab culture or any other techniques. So we are adding the enzymes and then reactants, whether amount of light is produced, and we are detecting using the luminators. So how much of cells are present in that sterile room area, we are detected by the how much of light is emitted. And then drug discovery. It is used in drug discovery, high throughput screening of drug candidate for cytotoxicity. And environmentals, as I said before, the sample is taken from the anywhere, biological sample or environmental sample. So monitoring the biotoxicity in wastewater and water for injections. So and then moving on to the advantages, it is highly sensitive. So even femtomolar range of the concentration present in the sample are detected, and no background noise. So here we are not using the light source. So this light source produces some background noise, but in this bioluminescence assay we are not using the light source. So it has low background noise and rapid result. Within a second we can obtain the result, and non-radioactive and safe. Compared to the radioactive assay, it is more safe. And limitations. So these enzymes, luciferase enzymes, are unstable due to temperature sensitive and inhibition by the sample impurities. If the sample contain any impurities, it can interfere with the results. So it is a major drawback of this bioluminescence assay. And signal decay. So it produce result at very second to minutes. But these emitted photons cannot withstand a few minutes, so it can decay rapidly and cannot differentiate the microbial species. So this bioluminescence assay cannot differentiate the microbial species. So in this session we are covered the bioluminescence assay. So hope you all understands. So next topic we are going to see is ELISA. So bye bye, thank you.